So we're getting into our next unit. Starting spring semester, we're going to be dealing with genes. Now, genes, we talk about genes all the time. And how genes are the directions that make us. So obviously it's not these kind of genes. But when we talk about our genes, it's the traits that make us who we are, what we are, and that kind of stuff. And so what we're going to talk about is what is called gene expression. How are the genes that we have, the genetic traits that we have, how are they expressed? How do we see those? When you express yourself, you are communicating or showing what you are thinking or feeling. So when a gene is being expressed, it is being shown or it is being expressed so that we can see what that gene codes for. What genes ultimately are, they're directions on how to make proteins. And so gene expression is also sometimes called protein synthesis. It's how do we go from a gene, the genetic code, the directions, to an actual protein. And the protein is what causes us to have that particular trait. And so we're going to be looking at gene expression, protein synthesis. How do we go from a gene to making proteins? Let's just remind ourselves of some basics. Just real quickly run through some basics over DNA. It's our genetic material. We got that. We know that. DNA codes for different genes. We have a lot of different genes in our DNA. And a gene is just a section. It's a part of DNA. That the DNA that we have 46 strands of DNA in every cell of our body. That the chromosomes, the strands of DNA, and a long piece of DNA can have multiple genes within it. We have thousands of genes within our DNA. So a gene is just a section of DNA that codes for one particular protein for one particular trait that we have. We've got to remind ourselves DNA, it's made up of nucleotides. The structure here, the nucleotide, the monomer of a DNA molecule, it has a phosphate group, sugar, deoxyribose, that's the D of DNA, deoxyribose is that sugar, and then it has a nitrogenous base, adenine, thymine, cytosine, or guanine. It has those 4 different bases. And they form this larger structure here. We get the 2 parallel strands going opposite directions with the bases paired up down the middle. They twist and they form this double helix structure of DNA. And what forms our actual genetic code is the order and sequence of these nitrogen bases down the middle part of the DNA. That's where our genetic code actually is. Within that, the sequence of adenine, thymine, cytosines, and guanines. All right. So DNA does 2 things, really just 2 things that it does. Sometimes what DNA does is it will replicate itself. It makes a copy of itself. That means DNA can make more DNA. That is a part of the cell cycle, and then ultimately it's creating new DNA so that when mitosis happens and we get new cells, that's in the S part of interphase, DNA can be used to make more DNA. That's one part. We talked about that back before the break. The second thing that DNA can do is that it can be used related to gene expression. And a gene expression, what we're talking about, is ultimately being used to build a protein, the protein synthesis. And the spot where all the work happens with that, the main thing where that's going on, is in the ribosome. And that's what we're looking at today. We are focusing on this lower part. DNA replication, that was DNA helicase and polymerase and all that for making a copy of our DNA. We're looking at this second part down here now, gene expression to ultimately make and create proteins. So if you just think of it basics, our DNA, stored in the nucleus, that's the information. And then that ultimately is going to get us in our body to making a protein. And I'm going to kind of use an analogy throughout these notes to keep in our mind. The DNA is not the protein. The DNA is just the directions. It's kind of like recipe. If I have a recipe box, there's a bunch of information in it, but it doesn't do me any good until I can make what it says that I'm needing to make, cookies. So think of DNA as like the information, the directions. The protein that is being made from our directions would be like making the cookies. All right? So sometimes it's referred to as the central dogma of gene expression. That's a big fancy way to say the big idea, the main big idea, the big concept of gene expression. And it's really 2 steps. The first step is that we have to take that information, we have to decode the information that is in DNA, and it gets copied into a new molecule, a new nucleic acid called RNA. And that copying is called transcription. And that's step 1. It just takes the information that was in the DNA and it copies it from the DNA into a new molecule called RNA. Transcription, that's step 1. Then step 2, the second step of the central dogma of protein synthesis, the second step is that the information that we copied into the RNA, we now have to decode that information, and we use it to actually get the individual amino acids, the monomers that form proteins, and we connect those to actually physically build the protein, which is a long chain of amino acids. That second step is called translation. Okay, so we start with DNA, and it gets copied, transcribed into what is called RNA. And then that RNA, the information on the RNA, is read and decoded into actually making the proteins. If we go back to my recipe concept, okay, DNA has all the information. Okay, that's like if I have a huge recipe book or a recipe box, it has a lot of recipes in it. Okay, when we're building a protein, we're just following one recipe. So like if I reach into that and I pull out one recipe, okay, I get a recipe for some chocolate chip cookies. Okay, that's like one gene. It is one set of information. And let's say I want you to be able to make those chocolate chip cookies. Okay, I'm not giving you my original recipe, but I will let you copy it. So you can copy my information onto your own recipe card. And so you copy that down. That's kind of like step 1. We are copying information from DNA, and we make a good copy. You copy everything down correctly. And then once you have your copy of the recipe, can you now go home, follow those directions, and then ultimately bake your cookies? Yes, you can. So if we think of it on just that simple little step, we have our original information. That information gets copied, and then the information on that copy is used to ultimately make the end product. All right? So the end product that we're trying to make here are proteins. Proteins are what form the structures within our bodies. They do all the work. Proteins are what make us us. I mean, they are the things that do all the work. I mean, just structurally, we've got to remember proteins are made by joining individual amino acids, monomers and polymers. The protein is the polymer. The amino acids are the monomers, the individual pieces that form. So this would be a long chain of individual amino acids. Each one of those little dots is one amino acid. And a protein is a huge chain of these things. But the DNA ultimately is a direction telling me what different types of amino acids to put in that big old long chain. Things that proteins do for us, and just remembering the jobs of proteins, the functions of proteins. They're structural. They form some of the structure in our body, the muscle tissue, hair, fingernails, the framework that our cells are on, the structural support of a lot of our cells in our body. They also are functional within our body. They work with our immune system as antibodies and hormones and enzymes and all that different kind of stuff. So proteins do all the different processes, all the different things going on in our bodies. So we have to be able to build lots of different proteins in our bodies for us to do what we need to do. And the guys that do this work, the ones that actually are helping to build and create and put together the proteins are the molecule RNA. And RNA ultimately, back when we were talking about biomolecules, we said nucleic acids, there's 2 types, DNA and RNA. And everybody was familiar with DNA, and we're like, that's what holds our genetic information. Yes. And then we said, we'll get to RNA later. We're later. RNA, ultimately, everything RNA is doing is working with making proteins, involved in protein synthesis. And then just structurally, RNA, it's similar to DNA in a lot of ways. It's made up of nucleotides. And so we know, that those nucleotides, it has a phosphate group, okay, just like DNA. It has a sugar. The sugar is different. It is a sugar called ribose. Which is why it is rRNA. That RNA actually stands for ribonucleic acid. DNA, the sugar is deoxyribose. That's the D, so it's a different deoxyribonucleic acid or ribonucleic acid. And then it has a nitrogen base, just like DNA does. But the nitrogen bases, there's 4 of them, but they are different. 3 of them are the same, cytosine, guanine, adenine. The 4th one is a nitrogen base called uracil. There is no thymine in RNA. Basically, uracil is a nitrogenous base that has almost just replaced thymine. There's no thymine, so anywhere in your mind, if you're thinking how they pair up with each other, cytosine and guanine still kind of work in combination. Adenine and now uracil. Uracil is that new guy who has kind of replaced thymine. Also, it is a single-stranded molecule. DNA had 2 strands going opposite each other. RNA is a single-stranded molecule. So if we're looking at them, we got this here. Here's our DNA, our double-stranded adenine, guanine, cytosine, and thymine. And our RNA is a single-stranded, still has the sugar and the phosphate backbone, but the bases just kind of stick out. They just kind of float out into space. And we have adenine, guanine, cytosine, and then we got this new guy, uracil, that replaces where thymine in our mind kind of would normally be. And then within RNA, there are 3 different types of RNA, and each one does a specific job critical in ultimately getting us from DNA to the actual protein. First type of RNA is called messenger RNA, and it's abbreviated mRNA. And what it is, ultimately, it is the copy of the DNA. It contains all the same information that was in the DNA, but it is now just copied over into RNA. This is like when you copy the recipe. This is your new copy of that information on the recipe card. It's created in the nucleus. The DNA doesn't ever leave the nucleus. The DNA stays contained inside the nucleus. But what happens is that we got to make a copy of the information in our DNA so that that information can then be taken out to the outside part of the cell to be used. So messenger RNA, mRNA, that's the first type of RNA. That is the copied of the original information. Then the second part is ribosomal RNA, or the ribosome, rRNA. It's an organelle. Ribosomes are organelles, and we said back last fall that the ribosome is the site of protein synthesis. This is where individual amino acids get put together. This is the location in the cell where the monomer amino acids get connected together to build and actually make the protein. So think of this as like the kitchen. Once you've gotten your recipe, once you've gotten your information, now you got to go to the spot where you can build the stuff you're making. You got to go to the spot where you can make the cookies. And so that's a location. It's the spot in the cell where you're going to build the protein. It's the kitchen at home. And then the last type of RNA is called transfer RNA, or tRNA. And what this guy does is it brings the individual amino acids that are needed to the ribosome. So when the ribosome is reading the information and saying, okay, what do I need? The transfer RNA brings the individual amino acids to it. And we'll get to kind of some animations that show how this works in a little bit. But I like to kind of think of this as like you got a little brother or sister that's wanting to help you make the cookies. You really don't want them to do any big work, but when you're reading the recipe and you say, okay, I need an egg, you tell little brother or sister, go get me an egg, and they get you an egg and bring it to you, here's an egg. And then you see the next thing, okay, I need a cup of flour, and you send little brother or sister, they go and get you and bring you the things that you need to build your product or to make the cookies. And that's what the transfer RNA does.